for those of you who don't know Jajia already, I think most do. Like Jajia and Xinghua were doing a sabbatical in SFIM. I think it was in 2018 or maybe 2017 and 18. This was a very productive time. I think they had 3-layer papers coming out of it, like 2 original papers spearheaded from Xinghua about somatosensory cortex and predictive coding, and 1 review article. And Jajia also had another paper, or maybe 2, I don't know, also combining 3 Tesla stuff. So I think now you are in Okayama University.
Yeah.
And continuing doing layer-fMRI with VASO, I believe. And those scanning, you don't have a 7T, right? You're using the Okazaki 7T, right?
Yeah.
And today he agreed to talk about his OHBM talk, which will be 10 minutes, and I will take the time. And
I will try to speak in German as well.
Feel free to go longer. I will take the time anyway, and then give you feedback. Okay, ready? And now.
So you can see my screen? Yeah, okay. Thank you, Renzo, for the introduction. So I have started my layer function study from 2016, and when I visit Peter's group, and I'm very lucky to meet Renzo there, and also Yuhui, Xiang, and all the members helped me a lot of. At that time, we have a hard time to guide scan slots, and we have to catch the good experimenter. And we have a very long journey to take our first data set, and after that, we go back to Japan. Renzo have already said we do some collaboration with Dr. Masaki Fukunaga from Okazaki. And now we also do some collaboration with Dr. Hanagawa from Kyoto University. They also have a human 7T scanner and animal 7T scanners. So hopefully we will have more time discussing in the future about what we are doing now on human layer function. We also do some animal studies. So, okay, I will start my OHBM talk, and it will be like 10 minutes. I will try to explain more detail about the studies. Okay, so first, my name is Jajia Yang from Okayama University, and my presentation title is Layer-Specific Finger Representation in Human Iris 3B. And this is a collaboration with National Institutes of Health in US and National Institutes for Psychological Science in Japan. I have nothing to disclose for this presentation. And first of all, I will show you some basic information of the somatosensory system. So there are 4 types of receptors in the skin that can be categorized into 2 groups. And the first group is named as slowly adapting receptors, SA, which are sensitive to the special feature of the object. And the second one is named as rapidly adapting receptors, which are sensitive to the temporal features. And both of SA and RA receptor will be received active at the same time and encode the information which contribute to our tactile perceptions. So when we touch an object, the information encoded by this receptor will be projected to the primary somatosensory cortex S1, which is located on the postcentral gyrus. And typically, the area have a body correspondence to a point on S1, which is called the somatotopic organization. So S1 can be divided into 4 sub-areas, 3A, 3B, 1, and 2. And the somatotopic maps in each sub-areas are different since the receptor field of the neuron in each area are different. So the previous animal study have shown that the receptor fields of iris 3B are small. So we expect a clear finger representation within iris 3B, and the receptor field of 1 and 2 become larger, so which reflect the convergent input from iris 3B. And also other errors. So in other words, the finger representations in area 1 and 2 may not so clear as in area 3B. Using functional MRI, we would expect to find a clear somatotopic mark in area 3B, and the map will be blurred in area 1 and 2. So in our previous study, we tried to map 4 fingers error in area 3-1 in human S1 at 3 Tesla. So we poke in subject's fingertip using stick in the scanner, and here is the map of one subject. So we can see even in area 3B, the finger activations are also overlapped, and it is difficult to isolate one from others. So this result did not match the receptive field in S1, as I have mentioned before. So the one possible reason for this result is the low spatial resolution at 3 Tesla. However, the overlap is still there when we look at the result at 7 Tesla with 1.5 millimeter resolutions. For example, in this study, in area 3B, the borderline between the fingers are not clear, and some of the voxels are activated by all fingers. So beyond the resolution issues, some sort of cross-finger interaction or feedback modulation in this area may be the factor for the overlap. So the finding from one recent study at 7 Tesla support this view by found that attention may modulate the somatotopic maps. So, but they did not discuss too much about the mechanism of this modulation. And layer-fMRI have the advantage to test the cross-finger interaction as well as the feedback modulation across these areas. So for example, in our previous studies, we found the finger input active the middle layers of area 3B, and the upper and deeper layers are more like activated by the predictive feedbacks. And we also found that prediction error feedback signal seems to modulate the activity in the deep layers of area 3B. So in this study, we are looking at the layer-space activity of each finger in these sub-areas of S1. And we try to find some insight into understanding the tactile hierarchical processing pathway in human somatosensory systems. And we designed this metal-free device, and you can see from this video. And when we push the one side down, and the other side will be raised up to poke the subject's fingers. And we use the multi-contrast VASO-BOLD sequence, which is a similar one that has published in Renzo Huber's paper. And the spatial resolution in depth is 0.71 millimeter, and the slash thickness is 1.8 millimeters. And the slash is perpendicular to the profound bank of post-central gyrus, and a total of 12 slices was recorded. And if effective TR is 3.49 seconds. And basically, this is an interleave acquisition mechanism. And each single visual image is interleaved with one bold image. And if we separate them, then we did pre-processing using AFNI, SPM, and FSL was used for statistical analysis. And here's the bold map for 4 fingers in S1 for one subject. So you can see the 4-finger activation were organized in a somatotopic manner in area 3B, but not show clear patterns in area 2. And here are the both clear color maps of index and middle finger for 10 subjects. And you can see some individual difference, but all subjects show the same finger arrangement along area 3B. And to see the finger distribution along columnar and layer dimensions, we have to define finger mask of each areas. And here is an illustration of S1, and the blue box as indicate or scan volume. After drawing the borderline of the green and white matter in area 3B, and we then calculate layer and using linear package. In each finger regions, we can find response in the middle layers from both VASO and bold signals. And we also found a clear finger representation in the middle layer of area 3B rather than the upper and the deep layers from the VASO profile. And in contrast, the bold activity showed mostly similar activity across all layers. And then to see the detailed data in area 3B and area 2, from layer and column direction, we then generate a new set of masks like this. And to give you a quick image, and here I plot data from one subject. So after repeat this process for all the subjects, and I normalize the data point in alignment directions. And here's the average, the bold profile. And I removed the error bar from the figure for visualization. And each small figure represents one data point covering all columns. And this figure confirms the clear activation for each finger in area 3B and like a double peak pattern in area 2. But the activation pattern seems the same across all the layers from this bold profiles. And here's the VASO profiles, and we found that 4 finger peaks in area 3B and the double peaks in area 2, and likely that more active this pattern are clear in the middle layers, but not all layers. And we are trying to expand our results using the previous animal findings for from the basic science side. Like so in this model, we know that from animal study, the first order information, sensory information will be arrived at the middle layer of area 3B, and then these signals will be projected to area 1 and 2, and further processing will be calculate how about the shape or something like the higher level processing in this across these layers. And we found a clear because the sensory input will be arrived at the middle layer and then spread to both up and deep layers. So this is the reason why we found clear finger representation pattern in the middle layers. However, to date, there's no direct evidence allowed that to model the layer-specific information across fingers and across these all these 3 areas. Almost we also missed we cannot record the activation in area 1. So it is super challenging to model the information pathway from 3B to area 2. And we are now trying to discussing the underlying mechanism of these layer-specific finger maps. And for more detail about study, please stop my poster, and I would be happy to talk with you. And finally, I would like to thank to all my wonderful collaborators, and thank you for listening.